Hello, my name is Chris Fiero, and I'm a student enrolled at Grand Canyon University in the Biology 515 course, or Physiology 1. And today's presentation is on the neuromuscular junction, which is the control of muscle contraction and extended onto relaxation. I will also extend it on into the sliding filament theory and understand what is happening with actin and myosin and in muscle contraction. So today's learning target is to be able to summarize the events involved in the control of skeletal muscle contraction and relaxation at the neuromuscular junction. And at the conclusion of this recording, I'll insert the video recording link to this slide. Before we begin diving into what is occurring, it's, I think, a good idea to have a couple of definitions. So the first one is the neuromuscular junction itself, which is the junction between an axon terminal of a motor neuron and the sarcolemma of a single muscle fiber. So here we have a muscle fiber. We have these axon terminals right here connected to the end plates of this muscle fiber. And then here we have our synapse. So here we have the sarcoplasm of our muscle fiber and our presynaptic neuron. And then some other definitions of note are axon terminal, which is that end of that motor neuron. Synapse, which is the space between the neuron and the sarcolemma. The sarcolemma, which is the cell membrane of a muscle cell. And then we have the motor end plate, which is the specialized region of the sarcolemma where that motor neuron connects, or where those axon terminals connect to that end plate or to that muscle fiber. Another thing of note that needs to be just understood prior to diving in is membrane potential. And that is the difference in electrical charge between the inside of the cell and the outside of the cell. In this case, we have our extracellular fluid or matrix, and then we have our sarcoplasm. Okay, and so at rest, the membrane is more positive on the outside, or in this ECF or extracellular matrix. And then here inside of the cell itself, it tends to be more negative. And this is referred to as resting membrane potential, and it's typically about negative 70 millivolts. Now, to initiate muscle contraction, we need an action potential to travel down the axon of that neuron and reach the axon terminal. And as it reaches that axon terminal, if you remember, it's connected to that end plate of a muscle fiber. Okay, so as that action potential is traveling down, it opens voltage-gated calcium channels at the axon terminals. And as that calcium diffuses into that axon terminal, it causes vesicles to release acetylcholine into the synapse via exocytosis. Okay, so acetylcholine is a neurotransmitter. So as it releases that acetylcholine into the synapse, it is able to bind to chemically gated or ligand-gated sodium channels on the sarcolemma. And as it binds to those channels, it opens them up. So now we have sodium channels that are open. And those sodium ions will then diffuse into that sarcoplasm and basically flip that membrane potential and be able to create a new action potential along that sarcolemma. So now that we've created or propagated another action potential on that muscle cell, okay, that muscle cell will in turn release calcium ions from the sarcoplasmic reticulum. And as those calcium ions diffuse into that sarcomere, they bind to the troponin-tropomyosin complex, or basically to the troponin. And that's when our sliding filament mechanism begins. Okay, so here we have actin and myosin. Okay, so myosin is in the blue and it's going to move those actin filaments, or it's going to slide them towards each other, towards that M line. Okay, and what happens as those actin filaments are moved towards that M line, we have contraction. So, as calcium ions from the sarcoplasmic reticulum bind to the troponin, um, those actin filaments are able to kind of like move and expose an active site. Okay, and that active site is where the myosin heads connect to that actin and are able to move it. Okay, so that's where the interaction occurs, is on that active site, between the active site on the actin and the heads of the myosin. So those myosin heads are energized and they're able to bind to those active sites on the actin, and that activity, or that process, is called forming a cross bridge. Okay, so once that cross bridge is formed, the energy that was stored in that resting myosin head is released. Okay, and as that energy is released, that myosin head is able to now pivot towards that M line, and the actin is pulled inward, okay, which means that the sarcomere is being shortened. And as that sarcomere is being shortened, that means that contraction is occurring. Okay, so once that myosin head is pivoted as far as it can go, if there's enough energy or more energy, and if there's ATP available, it will come and bind to that myosin head. Okay, and again, through hydrolysis, that myosin head is able to reactivate, okay, through the hydrolysis of that ATP. Okay, so that myosin head reactivates and it's re-energized, and the entire cycle repeats several times, and it'll continue as long as there's available calcium ions and ATP levels. Okay, so as long as those two things are true, and as long as what's occurring at the neuromuscular junction continues to occur, meaning that those sodium channels are able to kind of open and close and continue on with their process, we'll continue to have contraction. Okay, so basically, here it is in kind of graphical or picture form, which helps a lot of students usually. So here we have the myosin head. Here we have the actin filament. Okay, so this is the myosin here. This myosin head, once that active site's exposed, is able to bind to this actin here. Okay, and here, as we go down, we can see that this myosin head has moved towards that M line. Okay, so it's kind of bent or cocked itself towards that M line, and that is referred to as the power stroke. Okay, so as this power stroke is occurring, this actin filament is sliding towards that M line, which creates contraction. Okay, so as this actin filament is contracted, this myosin head needs to reset, and if there's ATP available, it is able to reset and continue that process to connect to that next active site on the actin and continue to slide towards that M line and continue the contraction cycle. At some point, the muscles do need to release, and the way this happens is that acetylcholine will diffuse away from the synapse and will be broken down by an enzyme called acetylcholinesterase. And as that acetylcholine is no longer available by those sodium channels will close, and sarcoplasmic calcium ion concentrations will return to resting levels. And then that means that the troponin returns to its original position and the tropomyosin covers that active site. Okay, so our troponin tropomyosin complex is back to what it was before. And then we have external forces that need to act on that muscle fiber to be able to stretch those myofibrils to their original length. Okay, so as the muscle relaxes, is able to return to its resting length. One of the things that I love to do with students is be able to kind of take notes at the same time with them because it helps to model that note taking process and it helps them visualize what should be on their own paper. So typically, what I'll do is go through the presentation as I have with you. And at the same time, I'll have this on a doc cam and I'll have a blank version for myself. For the students, I usually fill in an axon terminal and that sarcoplasm so that they're able to not have to draw and they're able to just kind of take those notes so we can move a little bit more quickly. But I'll give them this blank note sheet as just has the title, the definitions, the image, and then they're able to fill in kind of the rest as we go through. But I'll model this whole process with them and then I will color code it. And that's one of the things that I encourage all of my students to do is to color code their notes so that they're able to differentiate and understand what they're doing or what they're seeing. And then these are my references. One of the references I use is the textbook that we use in my physiology and anatomy class. And then the other is from our own textbook. And I will have this link uploaded as soon as possible and turned in. Thank you so much. Have a good night.